Okay, good morning, everyone. Not much to report on terms of assignments. All good? Any questions? Things going well? Okay. All right. So back to open challenges in the field of evolutionary robotics. We started last time by talking about one of the challenges of evolving neural networks, which is the competing conventions problem. We saw the NEAT algorithm, which is an attempt to solve this. NEAT has a lot of neat properties. One of them is that it allows you to evolve neural networks, which, as we'll see in a moment, you can then put back into robots for adaptive behavior. NEAT is very famous. HyperNEAT is another famous algorithm which builds on top of NEAT for a reason which we have not talked about yet, but we will talk about in a few minutes' time. We'll finish up our discussion of HyperNEAT in about half an hour, and then we'll move on to arguably the biggest problem in evolutionary robotics, and arguably in robotics in general, which is crossing the reality gap. No one has figured out a good way to cross the reality gap, despite several attempts to do so. Okay, before we get to the reality gap, back to NEAT and HyperNEAT, just to remind you of the problem, competing conventions. NEAT was designed to solve this by figuring out how to take two different things, like two different parental neural networks, and identify parts inside of those two parental structures that are likely to be performing similar functions. And the idea behind NEAT of identifying this is to keep track of the descent of those pieces. If those two pieces in two different neural networks have the same common ancestral piece or derived from the same common ancestral piece, they're likely to do similar things inside the neural network, and we're going to line them up to do it. Yeah? This has become sort of the standard way. The NEAT algorithm has become the standard way to evolve populations of neural networks to do things. We started last time by talking about HyperNEAT, which, as the name of the algorithm implies, builds on top of NEAT. So in the HyperNEAT algorithm, we're still going to be evolving populations of neural networks to do things. And we're going to mutate and sexually recombine these neural networks using the NEAT algorithm. What we just started to talk about last time is what exactly are these neural networks evolved to do in the HyperNEAT algorithm. We didn't even get that far. What we started in on at the end of last lecture was looking at how the neural networks in the HyperNEAT algorithm behave. We've seen a bunch of different neural networks in this class so far. You've probably seen lots of other kinds of neural networks in other classes. The neural networks that live inside of the HyperNEAT algorithm are known as CPPNs, not to be confused with CTRNNs. That's a completely different thing. CPPNs, the name stands for compositional pattern producing networks. And as we saw at the end of last class, they're called pattern-producing networks because they're neural networks that produce patterns. When we evaluate a CPPN, we define some space, and for the next few minutes, we're going to arbitrarily choose two-dimensional spaces because I can't draw in 3D. So we're going to choose a two-dimensional space, and we're going to construct different kinds of CPPNs and see what kind of patterns they produce inside this two-dimensional space. Okay, just a refresher where we were last time. Any questions about any of that from last time before we push on? Okay, so let's go through this by actually creating some CPPNs now and see how they work. As you can see in the cartoon up here, the input layer takes not sensor values or pixels from some photograph that the neural network is trying to categorize. CPPNs take as input coordinates, positions in some space. Since we're working in two dimensions, we'll do this with an X and a Y. And these can feed into an arbitrary neural network with arbitrary cognitive architecture. To start with, to build up our intuition for CPPNs, let's keep things really, really, really simple. We have two input neurons, X and Y, and we have one output neuron. And because this pen is failing, I would usually say black, but we'll go with gray this morning. Okay. Here's our minimal CPPN architecture. Let's add some synapses to this. Let's take one synapse and connect the left-hand input neuron to the one output neuron, and we'll give this a weight of 1. We'll keep things very simple. You'll remember back to our discussion about neural networks, we need one last thing, which is the activation function inside of here. When we take the weighted sum arriving at this neuron, we usually push the result of that weighted sum through a function. For now, we'll keep things simple, and again, we'll assume the identity function. So whatever the raw sum is that arrives here, that's what's going to come out of the output neuron. So far, so good? Okay, so let's now start to evaluate this CPPN. Let's take the coordinate of the origin, 0, 0, feed that into the input layer, and how much gray is the CPPN telling us to put at 0, 0? Hopefully you can all evaluate neural networks in your head by now. We're feeding in 0, 0 to the input layer. What's arriving at the output layer? 0. Don't put any gray here. Let's move to x equals 0.1, y equals 0. How much gray is the CPPN telling us to put here? Say that again.
1.
We've got 0.1 feeding in here and 0 here. 0.1, right? We take the presynaptic neuron, which is 0.1 at this time, multiply it by 1, drop it in here. Our CPPN is telling us to put 0.1 of gray here. Put a little bit of gray. How about 0.20? A little bit more, 0.2. You can see how this goes. I really need a different pen here. Let's switch to brown and hope that this works. A little bit better. Brown. Okay. We have evaluated part of the CPPN. Let's paint another row on top. So let's visit x equals 0, y equals 0.1. What does this CPPN spit out when we take that set of coordinates and feed it into the input layer? Nothing, right? We've got x equals 0 and y equals 0.1. The amount of brown we want to put here is 0. What happens if we put in 0.1, 0.1? 0.2, 0.1, 0.3, 0.3.1, 0.4.1, 1.1, comma 0.1. What's the pattern that this CPPN is going to produce in this row? Same as the bottom one, exactly, right? We get a horizontal gradient where we get increasing amounts of brown, and you can convince yourself that that continues. This particular simple CPPN will paint a pattern of increasing amounts of brown as we move from left to right. So far, so good? Okay, let's try another one. Let's create at random another CPPN. What pattern does this CPPN paint into this two-dimensional space?
Same thing, but rotated.
Same thing, but rotated. As there is more Y, as we move up in this image, this CPPN is going to dictate that we paint more brown into the picture. I'm really unlucky with my pens this morning. Let me see if I've got another one here. Aha. Black. Okay, great. All right. So let's try what's known as the inverse problem. Instead of giving you a CPPN and having you run that CPPN evaluation in your head to predict what the pattern is, now I'm going to give you a pattern and see if you can tell me a CPPN that will paint that pattern. Increasing amounts of black as we move up and to the right. can you construct a CPPN that would paint that pattern?
Two synapses, 1, 1.
Two synapses with a weight of 1 and 1. Okay, so far so good? Okay, so this is what CPPNs do. They paint patterns into a regular space. Why would we want to do this? We're almost at the motivation for CPPNs. Let's look at another detail of CPPNs. In the cartoon that you see over here, you'll notice that there are small little inset figures inside each of these 1, 2, 3, 4, 5, 6 neurons that make up this CPPN. What do you think those little inset pictures represent? The fact that they're being drawn inside the neuron is a very strong hint.
Activation function?
They're activation functions. In a CPPN, each neuron in a CPPN can have different activation functions. This particular neuron here is the identity function. So it may be hard to see from the back, but inside each of these neurons, there's a little small X and Y axes. The X axis is representing the value of the raw weighted sum that's arriving at that neuron. What's coming in? The activation function takes what's going in, it goes to that position on the horizontal x-axis, and then according to its activation function, it finds the corresponding y value at that horizontal position and spits out that value along any outgoing synapses. Yeah, so we're visualizing how the activation function for each neuron transforms its raw incoming signal into a processed outgoing signal. This one has a identity function. This one's got something that looks vaguely like a Gaussian. This one we haven't seen before. Here's an activation function that has some regular oscillatory repeating pattern over time. It's kind of reminiscent of a central pattern generator, something that produces a regular pattern over time, but it's hidden inside the neuron. Here's something that is definitely a Gaussian. When NEAT in the HyperNEAT algorithm is evolving populations of neural networks, the NEAT algorithm can set not just the number of neurons and which neurons are wired up and what the weights of those synapses are. It also encodes one additional detail that we haven't included in the genome before, which is the activation function associated with that neuron. In everything we've seen so far, all the neural networks, we as the human investigators usually picked an activation function. Now we're going to let HyperNEAT do so, or evolve activation functions into these neurons. Why might we want to do that? Let's go back to our example, our simple CPPN over here, and let's place a sinusoidal activation function inside. And let's go back to our simpler example of just a single synapse connecting the X input neuron to the single output neuron. What pattern is this CPPN going to paint in our two-dimensional space? A sign of the amount of black that we place, right? So maybe it starts with a little bit of black, more black, less black, more black, less black, more black, and so on. And then repeated as we go, yeah? Okay, why did the authors of HyperNEAT construct such an algorithm? Anybody have any intuitions yet?
Produces more diverse and complex patterns than using the same activation function for all.
It produces more diverse and complex patterns. It's true, it tends to produce regular patterns. So let's imagine a slightly more complex CPPN. Let's create it at random. And we'll randomly throw a sinusoidal activation function in here. We'll throw a Gaussian in here, and maybe the identity function into the final output neuron down here. We've got two input neurons, two hidden neurons, and one output neuron. We've randomly wired up the neurons. We've set some random weights. We've set some random activation functions. I don't know about you, but I haven't had enough coffee this morning to actually work my way through this and figure out what pattern it produces. But as you can probably guess, it's a mixture of regular gradations of more or less black in this space, and maybe there's a little bit of Gaussian noise here, or there's a little bit of Gaussian here, so maybe we put a little bit of black and then a little bit of more black in the center, and then less. You can imagine that what this CPPN does is to paint regular patterns, and it's composing these patterns together. Question?
That kind of was my question. Like, with all those patterns, it can kind of compose any...
Absolutely. So remember that no matter what kind of neural network we're talking about, a regular vanilla neural network, a CTRNN, a CPPN, ultimately all these networks are functions. They transform their input into output. CPPNs embody a particular kind or class of functions, which is compositions of coordinate transforms, which sounds intimidating, but you just saw that exactly, right? We're taking all of the coordinates, x, y, and z, and we're transforming them in a way that we're painting patterns onto them. And the way that we're transforming and painting these patterns is composed and composed and composed together. We get gradations of regular patterns across this two-dimensional space. I constructed this neural network at random, and if you've got time on your hands, take half an hour and code this up in Python and connect it to matplotlib, which you can use to draw pictures. And no matter how randomly you make the CTRNN, what you're guaranteed not to get is what you would do in the old days with an old television set. If you set it to an unprogrammed TV channel, you'd get white noise. Right? That there'd be no relationship between the color of this pixel and the color of the pixel next to it. Random CTRNNs produce non-random patterns. That's why they're called pattern producing, not image producing. CPPNs guarantee that you're going to get non-random patterns. You're going to get things that slowly change over space as we're painting into space. Everybody see that? Okay. I've held back the why. Why would you want to do this? Anybody have a guess for why you might want to do this?
So that you can take any randomly generated network that might not necessarily fit the body and make it fit into a body? Is that...
You're heading in the right direction. You are not a random collection of cells. You are a set of non-random patterns composed within non-random patterns, within non-random patterns, and so on. There is a gradation of physiological change across your 5 fingers. Fingers aren't exactly the same. They're very close to one another, but they also are slightly different as you walk from one finger to the next. The patterning in your arm as you march from your shoulder out to your fingertips. As you start to march outward, you see similar patterns of bundles of muscle and bone and ligament and tendon and so on. And that pattern gradually changes as you move along the body. Our insect brothers and sisters that are made up of a whole bunch of different segments. Now you can really see not just regular patterns, you can see almost sinusoidal or repeating patterns across the length of the insect body. There are segments, but then in these different segments, there are slightly different things going on. HyperNEAT was inspired not by biological evolution, but by biological development. The way that every single multicellular organism on this planet starts as a single cell, but as that single cell divides and starts to make a larger and larger and more complicated organism made up of more and more cells, What is going on during that process of development is fantastically complicated. And a lot of that developmental machinery, the stuff that's guiding development, the way you grow into a multicellular organism, is designed to place patterns within patterns, within patterns, within patterns. You are a composition of patterns all the way down. You are not some random collection of cells. Development is not saying, okay, there was one cell, now there's 2, now there's 4, now there's 860, now there's 1 billion. This individual cell, you go there. This individual cell, you go there. This individual cell, you go there. It's too complicated. You can't orchestrate this huge mass of cells and things going on by telling everybody exactly what to do. What you can try and do is influence big masses of them so that they organize into regular patterns across space and time. All the variety that we see out there in nature, if you look at every single multicellular species, there is fantastic diversity. But one of the things we all share in common is we are not just some random blob of cells. There is organization. It's different organization, but it's organization of patterns within patterns within patterns. Okay, that's this little cartoon here was the author's best attempt to try and visualize this. We've got a 2-dimensional space, and in theory, you could probably invent a CPPN that would paint, if we just imagine this is a black and white picture, it would paint a sinusoidal pattern along the y-axis, and they've shown this over here, and that might be composed with another part inside the CPPN that's painting black in a Gaussian manner across the x-axis, and that could produce something like you see here, something in which there is running up the y-axis, we've got more than less, then more than less, then more than less of black, and all of that black is bunched up at the middle of the x-axis. Everybody see that? Okay, so that could give us a regular body plan here. For some of these segments, we could have additional pieces of the CPPN that paint, again, some more repeating patterns, patterns with slight variations as well. And then it's also possible if you compose a bunch of Gaussians together, Gaussians can kind of squash or compress or focus one part of the CPPN to paint one and only one unique pattern at one and only one place in this 2-dimensional space. You can sort of get everything you need for creating something that looks vaguely like an organism. Repetition, variation on a theme, symmetries, and unique patterns as well, all within the same CPPN. So far, so good? Okay. All right, let's test our intuition about CPPNs. We now know why they were invented. If we evolve populations of CPPNs, we're searching the space of all possible networks that can paint all possible patterns. I could draw any unique pattern of black inside this 2-dimensional space, and with enough neurons and synapses, you, or at least a machine, could figure out a CPPN that would produce that pattern. If it was a random pattern of white noise, you'd need a very, 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 very big CPPN to do so. Smaller, random CPPNs tend to produce regular patterns in space. So when we're evolving populations of CPPNs, we're actually forcing evolution to spend most of her time in one part of the space of all possible patterns. It's the space of all patterns in which there is repetition, variation on a theme, and so on. If we created a fitness function that said draw white noise, evolution could do it, but it's got to move out of its comfort zone of painting regular patterns. If instead we had a fitness function that said paint oscillating waves of black from left to right in the 2-dimensional space, it would be much easier for HyperNEAT to evolve such a CPPN. This is something we haven't seen before. It is an evolutionary algorithm, but it's an evolutionary algorithm designed such that the genotypes, which are the CPPNs, tend to produce phenotypes in a particular part of the space of all possible regular repeating patterns in two-dimensional space. Make sense? Question?
That's similar with evolution and like nature. Like you think of all the different shapes of animals that there are.
Absolutely, yes. So in nature, mutations are random. So a random photon of light from the sun can hit some part of your DNA, unfortunately, and make a random change anywhere in the DNA, completely random. But the effect of that mutation, if it hits your sperm cell or your egg cell, depending on your sex, and that results in an offspring, the result of that mutation is going to be non-random. Luckily, your child is not going to be a random collection of cells. It's a mutation into a different regular pattern of cells. Absolutely. So biological evolution, mutations are random, but the effects of mutations are non-random. And that phenomenon itself has been evolved for very good reasons. If you need to survive long enough in the world to eat and reproduce, a good way to do so is to be a regular arrangement of cells, not a random arrangement of cells. Yeah, good observation. Okay, all right, so let's test our intuition about CPPNs. So far, we've kept things simple. We've been painting patterns of black and white into a two-dimensional space. In the cartoon example here, you can see that they're actually painting in color. How might you alter the cognitive architecture of a CPPN so that now it paints in color rather than just dictating amounts of black? What would we change?
You just have, in this case, 4 different output neurons for the different colors.
Okay, let's have 4 output neurons. What are those 4 colors? Or what are they going to? We're going to get numbers at these 4 output neurons. How are we going to interpret those numbers in order to paint in color?
I guess like the thickness of the pixel.
We could, the thickness of the pixel. We could pick whatever we want. Any other ideas?
Like RGB plus alpha.
Okay. For the non-computer graphics folks in the audience, what's alpha?
Opacity.
Opacity. Okay. So now we're going to set at least the input layer and the output layer. We're going to evolve with HyperNEAT populations of CPPNs, where each CPPN has 2 inputs and 4 outputs. And maybe there's one CPPN in the population that looks like this. And maybe elsewhere in the population, there's another CPPN that looks like this. And remember, we can have activation functions inside of all of these. We are now going to paint regular colored images inside here, where some of the pixels are going to have more or less opacity. Okay, great. What happens if we want to paint a regular pattern into 3-dimensional space? Let's stick with our colored example here. What do we need to change about CPPNs to paint inside a 3-dimensional volume?
Is it another input neuron?
Absolutely. X, Y, Z. And now we're going to define an empty 3D volume. And every time we evaluate some random CPPN, we're going to visit in a regular pattern. We're going to visit each 3-dimensional coordinate of that lattice in that 3D space. And we're going to paint a colorized image inside a 3D volume. Let's say we want to make a movie. We want a CPPN to generate a movie. How do we do it? This is part of the reason why HyperNEAT is so popular. It's very flexible.
You add another input for time.
Okay, let's go back to 2 dimensions. And we'll add a 3rd input, which is T, or actually maybe let's call this F, which is the frame index of our movie. There's one last thing we need to do. How do we evaluate this CPPN to produce a movie? We're going to make a movie frame by frame. Let's start by painting frame 0 of the movie first.
You have to evaluate it each time at each frame.
Each time at each frame. So we're going to create the 0th frame in the movie. So we're going to set this input neuron to 0, and then visit all the XYs in the frame, paint color onto all of those pixels in frame 0. We've now created the first frame of the movie. Now we set f equals to 1. We haven't changed the CPPN. It's 1 CPPN that's painting all the frames in the movie, and off we go. Make sense? Everybody see that? Let's say we want to now modify our CPPN to paint music. I give you an empty sheet of musical bars, and we want it to paint music onto it. It's 5 lines, right, for music? Okay. We've made images. We've made movies. Now we're going to make music.
Is the output neurons would each be like a note or a symbol that could be like a pause or?
Could be. There's different ways we could do it. Other ideas?
Again, the inputs are going to have to match the space that you're trying to fill.
Correct.
So it might need to reference each place on staff, but also temporally going forward.
Absolutely, right? So I'm not a music person. What are these called, these lines?
It's just
Spots.
Yeah.
Okay.
It's places on a stage.
Okay. Anyone watching this movie who's a music person, my apologies. Okay. Spots along the staff, right? So we're going to visit each spot, and then we might have a binary output neuron that tells us whether or not to place a note at that position or not. And we now need, again, pardon my ignorance, frame 0 is this. We visit the next point in time on the musical score, and now maybe it paints some other notes at that point in time, and we now have a CPPN that is generating music. Yeah? In all 3 examples, painting images, painting movies, painting music, we haven't said anything about the fitness function yet, like how good or how poor this music is, but you can imagine the last step in evaluating the phenotype. The phenotype is now the image, the movie, the musical score. We apply a fitness function for how good that image is, how good that movie is, how good that musical score is. Okay, this is not a music class, however, this is a robotics class, so you can probably guess where we're going. We're going to see an example, it's taken from this paper here, where the authors took the HyperNEAT algorithm and used it to paint behaviors onto robots. Okay. This is just to rephrase what we just said. The dimensionality of the hypercube, the 2D space, the 3D space, the 4D space, is determined by the dimension of the input coordinates. This is an important point. I want you to hold on to this. This is going to come back when we apply this to robotics. Okay, here we go. We, as always, want to evolve a quadruped robot to move as quickly as possible from left to right. So coming back, we've taken quite a divergence away from robotics. As usual, let's start by seeing if we can use this HyperNEAT algorithm to evolve quadruped gaits for a 4-legged robot in simulation. First of all, before we see how this was done, why might we want to do this? For those of you that have made it to assignment 10, you've taken this minimal 3-link 2-joint robot and replaced it with a 4-legged quadrupedal robot, and you can probably evolve a gait for this robot. Why would we want to go to a much more complicated evolutionary algorithm to evolve gaits for a 4-legged robot? What is it about HyperNEAT that might be of potential utility here?
Because it creates patterns, which is what
We remember our discussion about legged locomotion. We want patterns. What patterns specifically? Patterns in space, patterns in time. Yes, both, right? Remember the Ministry of Silly Walks? All of those walks were silly because they did, well, actually they did have a rhythm to it, but not the one we're used to, right? Random commands to the muscles in your legs are going to produce a silly, possibly dangerous gait, right? Not a good way to move. So what you're going to see in this experiment is some things that are familiar. We're going to evolve quadrupedal locomotion. The fitness function is the same as always, displacement along the x-axis away from the origin. But this particular evolutionary algorithm is going to bias the evolutionary process towards, hopefully, regular gaits. The problem, of course, is that the evolutionary algorithm is not acting directly on the motion of the legs of the robot. What the evolutionary algorithm typically acts on is the synaptic weights of the neural controller that controls the movement of the legs. So what do you think HyperNEAT is going to be used to do here? We know what we want it to do, which is indirectly bias search towards regular motions of legs. What do we want HyperNEAT to do? This little cartoon should be a strong hint.
Take probably a sine wave or something and kind of paint it throughout time so that each of the legs have activation functions that will happen to follow that kind of sine wave.
Okay, so maybe paint sinusoidal patterns through time. That's kind of what we want the legs to do, right? We want the legs to describe some rhythmic pattern through time, but we are not going to be painting in time.
I'm reminded of some of the diagrams you showed in previous classes of like when the feet were touching the ground.
Yep.
Some of those patterns. It's like we're trying to go from those patterns to movements of the limbs rather than the other way around with just mapping where the movements happen to land.
Exactly, so we're getting there, right? We're trying to work kind of upstream. We know where we want to get to. We want to get to a four-legged machine that moves with some rhythmic pattern. We don't know what that rhythmic pattern is. If we did, we'd just program it into the machine. We know generally that we want something that repeats for the motions of the legs, but there's still a bit of a gap here, right? The CPPN can't directly tell the leg, go here, go here. How are we going to close this gap? So one last step here. It's a little difficult to see here, but what you're looking at here is a neural network that's got a whole bunch of input neurons, and you'll notice that these input neurons have certain names associated with them. We've got a hidden layer in here, and we've got an output layer back here. So this should be starting to look familiar. This is a neural network that's going to control a quadruped. We've got an input layer with a whole bunch of sensors in here, a knee sensor or a knee angle sensor, touch sensors in the feet, angle sensors in terms of how much the hip is rotated forward and back, how much the hip is rotated inward and outward, starting to look familiar. The output layer here, next is the next torque sent to the motors. So a lot is looking familiar here, but we're missing, there's one thing that's not drawn here. What's not drawn?
Synapses?
The synapses. That's the hint. What is HyperNEAT going to do? We've seen HyperNEAT paint images, it's painted movies, it's painted music. Now, for this neural controller of the robot, HyperNEAT is going to...
It's going to paint synaptic connections underway.
Bingo, there it is. HyperNEAT is going to paint regular patterns of synaptic weights across the synapses inside this neural network. And this neural network is embedded in a 3D volume. That's what this cartoon is meant to show you. The hope is that if the HyperNEAT is painting regular patterns of weights across a neural controller for a robot, that is going to translate into regular oscillatory motions of the robot. That's the hope. It's not obvious that that's going to happen, and that was the goal of this project. Basically, this project embodies the following question. If evolution paints regular patterns of weights across synapses in a neural controller, will that translate into regular motions of the machine that's being controlled by that network? Everybody get it? You can probably guess, because we're talking about this paper, the answer is yes. And we'll see that in a moment. Okay, let's go through how HyperNEAT paints regular patterns into space. This can be quite confusing, so bear with me. Remember that we're running the HyperNEAT algorithm, which means we're evolving populations of CPPNs. Okay, we're going to take each CPPN, which is a neural network, and we're going to use that CPPN to paint synaptic weights onto a different neural network. So in this experiment that we're now talking about, there are two different neural networks at work. The former is painting patterns onto the latter. The CPPN is not controlling the robot. The CPPN paints regular patterns onto the network that controls the robot. So far, so good? Okay. They have chosen, as I just mentioned, to embed the neural controller for the robot in three-dimensional space, which means that every neuron in the neural network has a unique XYZ coordinate. That's going to be important in a moment. Yeah? In this picture, let's walk through this picture so you can see how this works. You can see that all of the 5 times 4, all of the 20 input neurons, all of the 20 sensor neurons, they're all embedded in a 2D sheet, which in turn is embedded in this three-dimensional space. That's this gray square here. The 5 times 4, 20 hidden neurons are embedded in a separate two-dimensional space, two-dimensional sheet. And then finally, they've got 20 motor neurons, but as you'll see in a moment, they only need 3 times 4. They only need 12 motor neurons for their quadrupedal robot. So we've got 20 sensor neurons, 20 hidden neurons, and what did I just say? 12 motor neurons. Again, all of those neurons have their own unique XYZ coordinate. So far, so good? Okay, let's go back to the sensor layer for a moment. Inside the sensor layer, they have arranged each row of sensors is dedicated to one of the 4 legs that makes up the quadrupedal robot. I haven't shown you the quadruped yet, but you can probably guess how this goes. Okay, so leg 1, let's just assume this is the front left leg of the robot. There are 5 sensors inside that leg. There are 3 of these, or sorry, 3, what is it? 1, 2, 3. 3 of these sensors are proprioceptive sensors. The first proprioceptive sensor in the front left leg indicates the angle of the hip, how forward or back the leg is at the hip. The leg is attached to the main body at the hip. How much is that leg rotated forward or back? That's a floating point number. The second proprioceptive sensor in the front left leg reports how much that front left leg is rotated inward towards the body or outwards away from the body. How much that leg is flexed inward or extended outward. Third number is the angle of the knee. So we've got 2 proprioceptive sensors sitting in the hip and 1 proprioceptive sensor sitting in the knee. Same thing for the front right leg, back left leg, back right leg. So far, so good? Okay. Fourth sensor in each leg, pretty straightforward. It's a binary value, whether or not the tip of that leg is in contact with the ground or not. Sorry, I misspoke. There are not 5 sensors in each leg. There are 4 sensors in each leg, 4 times 4. And then they snuck in some additional 4 sensors here. Yaw, pitch, and roll. It's not drawn very well. These are not associated with any one leg. They're associated with the main body. They're obviously measuring the yaw, pitch, and roll or orientation of the main body. You can also sense yaw, pitch, and roll as you move about through your day. How? Thank you. Right, inner ear, right? So our robot has an inner ear. And then finally, the 20th sensor neuron is emitting a sinusoidal pattern. This should look familiar. What is that neuron simulating?
Like 1 hertz rhythm walking.
Which is called, in us, in your spine, central pattern generator, right? In case that's useful. So, not that we've seen this before. The only innovation here is they're embedding this in a spatial embedding, in a 2-dimensional plane. These are somehow going to be wired up to the hidden layers. We'll assume for our purposes that each one of the 20 sensor neurons sends out a synapse to connect to one of the hidden layers. So we're going to have a fully connected sensor to hidden layer. So far, so good. So each of the 20 sensor neurons connects to each of the 20 hidden neurons. We have 20 times 20, 400 synapses that are connecting the sensors to the hidden neurons. So far, so good? Question?
So, okay, so they're not trying to paint any particular hypothesis onto the weights. They're just trying to create some sort of bias to make it so that the guesses that it has in terms of connecting all these things together are not start, stop, crazy.
Correct. Exactly. So again, they haven't drawn the synapses here so that you can paint with your imagination what those synapses might look like. So imagine we drew all 400 arrows coming out the back of these and into the front of these. And imagine we drew the lines of those synapses thicker if they were a stronger weight, or thinner if they were a weaker weight, a weight closer to 0. If we did this in the way you're doing it in the assignments, you just see some random collection of thick and thin lines. But with HyperNEAT, no matter what CPPN we create that paints weights onto these synapses, if we looked inside and across these 400 arrows, you'd see regular increases and decreases of thicker and thinner lines. That would visualize the internal regularity of synaptic weights. Okay.
Why embed this neural network in a 3D space? Why not 4D or 5D or 100 dimensions?
Great question. Because it's harder to think about it that way. By embedding it in a 3-dimensional space, we can at least try and draw the cognitive architecture, what this neural network looks like. There's nothing that says we couldn't take all of these neurons and embed them in a higher dimensional space and do exactly the same thing. Maybe it's advantageous, who knows? That sounds like a great final project idea to me. You just wouldn't be able to draw what that neural network looks like easily. You could also embed all these neurons in a 2-dimensional space and paint regular patterns in there as well. Is that better or worse? Who knows? Hard to say. They just picked 3D. Okay, let's keep going. So we've got 400 synapses going from the input to the hidden layer, and imagine we fully connect all these 20 hidden neurons to these 12 output neurons. We've got 4 rows again. Each row allocates motor neurons to that leg. So these are the 3 motor neurons assigned to the front left leg. These are the 3 motor neurons assigned to the front right leg, and so on. It does not say on this picture where those 3 motor neurons go in the leg, but given our description of the sensor neurons, you can probably guess. where do the 3 motor neurons go inside the leg?
The hip forward, backward, the hip in out, and the knee.
That's exactly it, right? So here's our hint. We know that the leg can rotate inward and outward at the hip, and the leg can rotate forward and backwards at the hip, which tells us that there are actually 2 hinge joints there. It is not like our knee, it's like our shoulder or our hip, forward and back, and inward and outward. So we've got 2 motor neurons that are controlling those 2 degrees of freedom in the hip, and then the third one is controlling the knee. Got it? Okay. So that's the neurons. We know the distribution of sensors. We've got full connections between inputs to hidden and hidden to output. Finally, thanks for your patience. Now we can do the fun part. Let's imagine a CPPN that's going to paint patterns onto each and every one of the synapses. So when I introduced you to CPPNs, we walked through positions in a 2-dimensional space. And at each point, we obtained a single floating point number, which was the amount of black to place at that position. Now, we've got synapses embedded in a 3-dimensional space. We're going to visit each synapse in turn and take the coordinate, the position of that synapse, feed that position in, and we're going to have a single output neuron. Actually, let's draw this. We've got our synapses in 3-dimensional space, so we need the XYZ coordinate of a synapse, and then we've got a bunch of stuff going on inside, and we have one output neuron that we're going to call W to remind us that the value arriving here, this floating point number, is going to paint that weight onto the synapse at that position. Same CPPN, we're going to call that CPPN 400 times to paint 400 weights onto the 400 synapses between the input and hidden layer. Take the same CPPN and paint all weights onto all the synapses that are going from the hidden to the output layer.
So the neurons are arranged in XYZ space.
Yep.
So this would generate one value for each neuron, but you need one value for each synapse.
Yep.
And so each individual neuron needs, say, 20 values, not a single one.
Okay, a good observation. So deliberately I'm making things simple, and it's too simple. It's not going to work. But we're going to see, I want you to see if you can see exactly why it's not going to work. We can, if we take this synapse, if we take a synapse that is in 3-dimensional space, it starts at some neuron that has a unique XYZ coordinate, and it ends at another neuron that has a unique XYZ coordinate. The center of that synapse also has some XYZ coordinate. Everything in here, all neurons and all synapses, are embedded in a 3-dimensional space. So when I said we're going to take the CPPN and visit each synapse, let's actually visit the centroid of each synapse. We're dealing with this neural network geometrically now. We're going to visit each centroid, take the XYZ coordinate of the centroid of that synapse, and get back a weight from the CPPN, 0.4, let's say. So we're going to get floating point values for all of these synapses, but as Nate has started to intuit, there's something wrong with this process. What is it? Not necessarily wrong, but there's something that's limiting us here. We're using CPPN, so we know it's going to paint arbitrary, regular patterns across all of these synapses, which is good. That's what we want. But we're actually hobbling ourselves here a little bit. So there's something that's holding us back.
Some synapses have the same centroid.
Thank you. Exactly. So let's look at this sensor neuron up here, and visually, I want you to paint a synapse that goes from this neuron to this hidden neuron. Imagine now we visit this neuron, and imagine the synapse that connects it to this neuron. Those two synapses which both are embedded in three-dimensional space, are going to have the same centroid. So tell me about the two weights for these two synapses that share a centroid. They're going to get the same weight, right? No matter what no matter what CPPN we apply to this network, it's going to be forced. It has to assign the same weight to sets of synapses that share a centroid. Now, this might not actually even be a problem. We might be able to evolve neural controllers for a quadruped such that it exhibits regular patterns over time. But if you're mathematically inclined, something doesn't quite sit right with this. It would be nice to roll back this unnecessary constraint. How might we do it? There's different ways we can do it. There's one particular way that the authors did it. They didn't like this. They made one change so that they made one change to the cognitive architecture of the CPPNs so that it could paint different weights on synapses that share a centroid. How do you think they did it? Any ideas?
Instead of looking at the centroid, look at the point just a little bit along the line to show you the vector.
Yeah, you could. So instead of the centroid, maybe we take a point 3/4 of the way along the synapse towards its postsynaptic neuron. That would work. That's not what they did.
You could still I guess maybe. You could still end up with them like crossing.
Maybe, yeah, it depends. Yeah, you'd have to work it out. You probably could pick a point at which they don't. If you went in and actually measured things, and like all good scientists, these scientists who I know well, they were a little lazy. They didn't do it that way. They introduced a little mathematical trick so that they didn't have to measure anything. They could be pretty secure that the CPPN would feel free to paint unique weights, even if synapses shared a centroid. Any ideas?
They introduced a random number to the one.
You could introduce a random number, but that makes things hard for evolution, right? Because now we're changing things every time evolution goes to evaluate a CPPN. This is not trivial, so it's okay. I'll give you a hint. Cam mentioned a few minutes ago, why not embed these things in higher dimensions? Okay, you're getting very close now. So this neural network, the network that controls the robot, is trapped in three-dimensional space, but they started to add input neurons to the CPPNs that were painting patterns onto this. So this gets a little bit tricky. We're juggling two different kinds of neural networks, the CPPNs and the robot neural controllers, and now we're going to start juggling dimensionalities. The neural controller is embedded in three-dimensional space, but the CPPN is going to paint in higher dimensional space. You mentioned one idea, which could be we're going to add some sort of neuron or synapse index, in very much like we saw in the movie example, right, where we had a frame index for frames in the movie. That's actually one way you could do it. That's not the way they did it. They added 3 additional input neurons, which means they now have a CPPN that's painting regular patterns inside a 6-dimensional volume. I hope you had your caffeine this morning. Okay, there's the hint. 3 additional input neurons. What are they?
So it's the first 3 are the starting neuron location, the second 3 are the target neuron location.
Absolutely. Okay, so here we go. Remember that every synapse has a presynaptic neuron, the place where the synapse originates, and every synapse has a postsynaptic neuron, which is the neuron at which that synapse terminates. We've got pre and post, that's 2 P's, so that's not going to work for us. Let's go for pre, R is in pre, and O is in post. So they're going to visit each synapse in turn, and they're going to feed into the CPPN the X, Y, and Z coordinate of the pre synaptic neuron's 3D position. And they're going to supply the x, y, and z coordinate of the posed synaptic neuron at which the synapse terminates. Okay, let's summarize. We're going to evolve neural controllers for a simulated quadrupedal robot. That neural controller looks more or less like we're comfortable with. The neural controller of the robot is embedded in three-dimensional space. We're going to evolve not the neural controllers. We're going to evolve populations of CPPNs. So the genotype in this evolutionary algorithm are CPPNs. Each genotype is going to paint synaptic weights onto this neural controller using 6 dimensions. Once the CPPN has finished painting the synaptic weights onto the neural controller, this neural controller is the phenotype of the CPPN. The neurons and synapses inside the CPPN is the genotype, the stuff that HyperNEAT is going to operate on. The phenotype is the neural controller to which we're going to assign fitness. So far, so good. We're putting everything together here. So we've got a population of CPPNs. We've taken the first CPPN, painted weights onto the neural controller. We let the simulated quadruped be controlled by that neural controller. We measure how far that simulated robot travels from the origin, which gives us back a floating point value, how far it traveled. That's the fitness of that CPPN. Go to the second CPPN, it paints neural control onto the quadruped, and rinse and repeat. Got it? Emily?
So 6 dimensions outbreak 3 dimensions.
The CPPN is painting regular patterns into a 6-dimensional space, which is then interpreted back down into 3 dimensions, right? This is the advantage of higher dimensions. When you work in higher dimensions, things tend not to overlap as much, right? The lower dimensions you go to, whatever it is you're working with, sets of synaptic weights, mathematical structures, you tend to get a lot of overlap. You can pull stuff apart by going to higher dimensions. It's a very, very common trick in mathematics. Hopefully here you see exactly why we're doing it. Okay. Okay, so here's my little cartoon of how this works. We're feeding in the coordinates in 6-dimensional space of each synapse, passing it through a CPPN, and it gives us back the weight. This is kind of the key of this whole thing. Okay, okay, we already actually said this. You can fill this in on your own. Okay, so here we go. Remember that the question the authors wanted to answer was, does HyperNEAT make it easier for evolution to discover regular gaits for the quadruped compared to other things? So what they did was to create the other thing. This is called the control, the thing that's missing HyperNEAT. They called this FT-NEAT, or fixed topology NEAT. This is a little confusing. I don't really like this control, but this is what they use, so here we go. They used the same neural controller, the same neural net as in HyperNEAT. They used this thing, but now that allowed for mutation and crossover, but not the addition, removal of neurons and synapses. So they're sort of fixing this. They're removing all of the good stuff about HyperNEAT, the ability for HyperNEAT to paint regular patterns in space. I think just you'll have to trust me on this. If you're interested in the details, you can go and read the paper. But we're going to look at 2 different algorithms. HyperNEAT is the algorithm we just talked about. The control is everything is exactly the same, but they've removed the ability of HyperNEAT to paint regular patterns in space. They've hobbled HyperNEAT in a certain way. It's kind of collapsed back down from HyperNEAT to NEAT. I think we won't go into the details, just have to trust me on this. Okay? All right. Okay, let's watch some movies. Okay. This is the best, most fit gait or the most fit neural controller they got from the control experiment. The evolutionary algorithm that could not paint regular patterns of regular patterns of weights across the synapses. How's it doing? It's okay. There's a little bit of rhythmicity here, but not quite. If you watch any one leg, you'll see that it might be in phase. It might be moving with one of the other legs, but then kind of falls out of phase with it. If this robot could sweat, it'd be sweating, right? There's a lot of exertion here. There's a lot of points in which the leg hits rotated forward, which stops motion. It's lost its momentum. This is a silly walk. This is not a very efficient way to move. This is the best neural controller they got from the control experiment. That was evolving sets of synaptic weights for this neural controller, but no regular patterns within the neural controller. This is a little confusing. I'll explain this as we watch it. This is the worst evolved gait, which actually means the worst of the best. So they evolved with HyperNEAT. So they're evolving regular patterns of weights across the neural network. They ran HyperNEAT once and got the most fit neural controller out of that evolutionary algorithm. Then they went back and created a different random population of CPPNs and ran HyperNEAT a second time and took the most fit neural controller out of that second run. Now they've got two highly fit neural controllers, ran HyperNEAT again, third time, a bunch of times. So they had a set of champions. These were the most fit individuals from a whole set of independent evolutionary runs. And among all of those champions, they took the worst one, the worst champion. It's a little confusing, the worst of the best.
It seems like it wouldn't be that hard to like just directly program a robot with just like do
Yes.
this with all of its legs at once.
Yes, absolutely.
Like, isn't the point that we're trying to get it to do stuff that's harder than we can design ourselves?
Yes, absolutely. For this simple robot, you could probably go in, and you're all good enough roboticists now, you could probably pre-program it to do so. You might remember the nonaped from the beginning of this course, that weird metal tubing type robot that had 9 legs that you also wanted it to move. In that particular robot, there were 12 motors, so you'd have to control 12 motors. Give that a try. See if you can sit down and figure out a way for that thing to move with a regular gait. It doesn't take a lot more mechanical complexity. I can put a few more joints and links on here where it suddenly becomes very difficult to figure out how to program a regular gait for it, especially if it's moving over uneven terrain and it's being buffeted by wind and somebody comes along and drops a 200-pound backpack onto its back. Not so easy to do in that case. So you're right. This demonstration is maybe a little bit underwhelming because you could probably do just as well. You can try and run this on a more complex robot and see how things go. Okay, so this is the best of the worst. How is this one doing? Got a regular gait, right? So it works. You can actually, you can use CPPNs to paint regular patterns of synaptic weights inside the neural controller of this robot, and you tend to get regular patterns of motion. We've spent most of the morning talking about how synapses are tuned inside of a maybe not complicated neural network, but getting there. What do you think's going on in your own head? You tend to do most things with a regular pattern arrayed over space and time. It's not quite known yet about synapses, but neurons definitely seem to have regular patterns inside our central and peripheral nervous system. It looks, on first pass, it looks like some random jumble of neurons. Most of the last 100 years of neuroscience and neural physiology, dissecting neural networks, has been to answer that question. Is it actually just a random tangle of neurons and synapses? The answer is, at least in the way it's wired up, absolutely not. There are definitely regular patterns, and not just regular patterns, but compositions of regular patterns. You get teeny tiny little neural modules that are made up of just a handful of neurons that are wired up in a particular way, and that little neat packet seems to be replicated into a larger structure that's made up of these smaller structures. Those larger neural modules are again replicated in the brain. Some of them are replicated with variation. You see pattern composed within pattern, composed within pattern, within biological neural systems, which suggests, although it has not yet been proven, that that tends to bias our form and function towards forms and functions that unroll over time and space in regular ways rather than random ways. So maybe HyperNEAT, which was only designed to help with AI and robotics problems, might suggest or give us ideas about why biological nervous systems are arranged in the way that they're arranged. Nate.
The cool factoids, like the sensory and motor neurons in the brain are actually spatially organized.
Yeah, that's right. The organization is not necessarily non-intuitive. It's, as you mentioned, the sensory and motor parts of your brain are arranged in a regular pattern, which actually matches more or less one-to-one your actual physiology. And actually, we'll see this. We're going to see what's called the motor homunculus in a few weeks in this class. We'll come back to exactly this point. Okay, I think that was all the fun movies I had for today. The best of the best? Well, if this is the best of the worst, you can imagine the best of the best is even better than that. Okay. All right. We've got a grand total of 2 minutes left. So all I want to do today is just introduce the next problem we're going to try and solve, which no one has solved yet, the reality gap. We're going to see, I guess on Thursday, we're going to see an evolutionary robotics experiment that dates all the way back to the beginning of evolutionary robotics, which was in 1994. In that paper, they introduced this term, or they introduced this problem, the reality gap. We're going to see that they simulated some robots and then tried to transfer them to reality, and it didn't work so well. That was 1994. In 2018, this lovely company here came along and said, listen, there's this problem in robotics that none of you have identified yet. We're going to call it the domain randomization problem. And we are the one and only company that's going to be able to solve this problem. All great problems are reinvented or rediscovered every 20 years or so. So this is a very big and very real problem that some very big and powerful corporations are trying to solve. It all started with a modest evolutionary robotics paper from 1994, which we'll talk about, not on Thursday, we'll talk about it next Tuesday. You have a quiz due tonight. Some of you are working on assignment 9, some of you are working on D4. See you then.